Hey, hello guys, welcome, and the name is Karim. Today we are going to see part 3 of molecular basis of inheritance. In this lesson already we have covered part 1 and part 2. Now it is part 3, so we will be proceeding with genetic code. What is a genetic code? Genetic code is a code that is given by scientists in order to identify an amino acid. Okay? It is a code that is a triplet, okay, that has triplet amino bases. That means, for an example, one code, okay, one genetic code will be having 3 different amino acids, or it can be same amino acids also. So their examples are like AUG, GUG, GUU, GAG, like that. Their examples vary. For example, see, AUG codes for methionine. GUG and GUU codes for valine only. These two are coding for same amino acid only, which is valine, even though they are different. Even though G and U are different in this place, but since they are in the third position, of the genetic code, these positions are said as obal positions. Okay, so these positions are said as obal positions. What are obal positions? These are flexible positions where even though if you change their amino acids, they will be coding for the same amino acid only. Even though if you change their nitrogenous bases, but they will be coding for the same amino acid only. Okay, like you have GAG, which means glutamic acid, GUG, which means methionine. So like that it is there, right? Totally there are 20 different amino acids. Okay, and there are 4 different nitrogenous bases. We are going to see only in RNA, not in DNA. Whatever I'm saying, right, that is all you have to relate with RNA only, not with DNA. Okay, so first this George Gamow is the physicist who found that a genetic code should have about 3 different, nitrogenous bases. Okay, so a genetic code should have only 3 different nitrogenous bases. This is found by George Gamow, who's a physicist. Okay, so yeah, so if a genetic code is having 3 different nitrogenous bases, then that means how many genetic codes can be formed? About 64 genetic codes can be formed. First, what they thought is there is only 2 different nitrogenous bases, or 2 nitrogenous bases can be there in a genetic code. But when you are having only 2 different or 2 same nitrogenous bases in a genetic code, then that means there will be only 16 genetic codes, but totally there are 20 amino acids. So that means that is impossible. So what he said is there are totally 3 different nitrogenous bases in a genetic code. So these are theirs, these are those. Okay, he used permutations and combinations. Okay, so these are those 20 different amino acids. What are they? Let us look at that. So alanine, cytidine, cysteine, then aspartic acid is there, glutamic acid is there, phenylalanine is there, glycine is there, histidine is there, isoleucine is there, lysine is there, leucine is there, then methionine is there, asparagine is there, proline is there, glutenine is there, arginine is there, serine is there, threonine is there, tripopropane is there, then triosine is there, and valine is there. Okay, totally these are those different 20 types of amino acids. Well, if you check it out, here I have given in the alphabetical, not I, the person who created this image has given in the alphabetical order. B is not there, like that, some, only a few, okay, like 4, only 4, sorry, only 6 is missing. Other than that, all other alphabets are there. Other than 6, all other alphabets are there. Okay, next is, now we will be checking out. See, over here in the first slide even I said that these 2 code for the same amino acid. Like that, there are still some other examples. Let us look at that. UUA, UUG, CUU, CUU, CUC, CUA, CUG. Okay, all these code for the same amino acid, that is leucine. Okay, that is leucine, which is degeneracy. Okay, that is degeneracy. See, here you are having 3 different scientists. Okay, totally you are having 3 different scientists where they have done a lot of experiments. First is Har Gobind Cohen, who is actually chemically, he synthesized mRNA. Okay, chemically synthesized mRNA. mRNA. Okay, he used only two different things. What are they? They are copolymer and also heteropolymer. Copolymer and heteropolymer. Gobind Cohen, one of the important scientists. Most important is this fellow, Sir Verno Okoya, some sort of name, I think Chinese name. Okay. Then, what did this Marshall Nierberg, one of the most easiest and simplest name, and also his research is very simple. He synthesized protein. Okay, proteins. He synthesized mRNA. Here he synthesized proteins. Okay, proteins in the lab itself, cell-free. In lab even he was able to synthesize proteins. Then he is the important fellow because he is the one who produced synthetic RNA polymerase. For synthesis of protein also, and also for synthesis of mRNA also, we want RNA polymerase. But they all used natural RNA polymerase. The RNA polymerase that is available in the nature itself. I think they all would have extracted that from the E. coli itself. Okay, so from E. coli itself they would have extracted it. Yeah, so enzyme. Next is this one. Yeah, this is known as degeneracy code. And also there are some codes which code one and only for one amino acid, like AUG. Okay, so like that's for first page. That means page number 95 has got completed. Still only some topics are there, we are going to complete that also. So degeneracy. Next is we have to read, so rules of this codon. You have to read these codons. No, this is not that good. I'll write here even. Okay, you have to, no punctuality should be there. No punctual uses. No punctual uses. That means what? That means, for an example, if you are writing, I will be taking methionine itself, AUG, there should be no commas, or there should be no exclamatory marks, or there should be no full stops. Like this, nothing should be there. Okay, if AUG is there, right? If AUG is there, now then that should be read as AUG. When there should be no stopping. But it does not mean that you should not put comma between two different amino acids. You can put comma between two different amino acids. That means AUG, comma CUU. Okay, so like that you can put different punctual marks. Okay. Sorry, that's my father. Okay, so coming to this case itself, we cannot use any punctual marks. Okay, they have to be read all along. Then any code that is actually used only once, for an example, they are known as specific codes. Specific codons. What are they? They are known as specific codons. Only once they can be, like for an example, AUG. AUG can be said as the specific code. Okay, so then after that, after this AUG, then what else I can give? We are also having universal language. Yeah, this is a universal language. Okay, so this is a universal language. That means if AUG is there in this universe, then another universe also, it means AUG means methionine only. Okay, just kidding. That means in any different organism. Okay, in any organism, AUG means methionine only. Okay, there is no different name for that except in mitochondria and chloroplast. Mitochondria and chloroplast. last. Okay, then also initiator codon. Yeah, so this AUG is there, right? Do you know why I just give this much stress to this AUG? Because this is the initiator codon. You all must have heard the word called as translation. Okay, translation is the process by where a RNA or mRNA is converted into proteins, right? So for that, this is the initiator. If AUG is there in mRNA only, that can be converted into proteins. If in case there is no AUG, then there won't be any proteins at all. Next is stop codon is there. Yeah, like this. This is the initiator, or this is what the start codon. You can see this as the start codon. We are also having stop codon. What is the stop codon? See, we have UAA, then we are having UAG, then we are having UGA. What does it mean? It means that does not code for any amino acid. These three are the stop codons. Okay, these three are the stop codons. Okay, but the thing is they don't have any name because they don't code for anything at all. For an example, if this is what the mRNA is, here this is what the initiator is. Okay, this is what the initiator is. That means here AUG will be there. Okay, so the translation process happens. Proteins are being created, and here they will be stopping. Here, for an example, if there is UAA or UAG, or if there is UGA, okay, if there is UGA, no matter what they are, if there is this any three, okay, amongst this any three, any one genetic code is over here, then that means the total translation process itself will be stopped. So these three will not code for anything. So totally there are only 61 codons out of 64 codons. That is pretty good mark. 64 codons out of 64 codons that can score. Okay, that can not score. That can produce amino acid. Okay, yeah, so I'm pretty satisfied with till now. Okay, next is coming to the structure. We are having tRNA structure. Okay, this is not in our NCERT book. I think, I don't know whether is this available. Okay, yeah, coming to those 64 different codons. See, first, this is the letters which are written over here in this box, right? That has to be written first. For an example, if there is U, then see U only is in the beginning. We are all, oh wait, oh shit, I didn't mean to clear that. Okay, now it has got clear. Okay, see, U. If U is here in the first, then here also will be U only in the first place. If C is here in the first beginning, then here also C only will be there. If A is here over here, then here also A only will be there. If G is over here, then G only here also will be here. Okay, so that is what it actually says. Throughout this entire box, first letter will be only U. Throughout this entire box, first letter will be only C. Throughout this entire box, first letter will be A. Throughout this entire box, first letter will be G only. Also, see, this is what the second letter is. Okay, second letter should be, this is what the second letter is. So throughout this entire box, second letter is U. Throughout this entire box, second letter is C. Throughout this entire box, second letter is A. Throughout this entire box, second letter is G. Also, these are those third letter boxes. So throughout this entire box, it's not like that. In this first row, it is U. In this second row, it is C. In this third row, it is A. In this fourth row, it is G. Like that, it will be continuing. I don't know why am I dancing. Okay, so this is what the genetic code table. So now we are going to mark a few. I'm going to mark AUG. So A, U, G. See, this is where the start codon is. Okay, then there are these three stop codons. Okay, they have even marked for us. UAA, UAG, then UGA. First letter has to be U. Okay, first letter must be U. Then after that, second letter should be A. Okay, second letter should be A. Third letter should be G. So, which means the first letter should be U. Okay, second letter should be G or A, and also third letter should be G or A. First letter should be definitely U. Second letter should be, it can be G or it can be A, and also same applies for the third letter. Wow, very easy, right? Oh God, I can't erase over there. Okay, so this is what it means. Okay, then coming to the structure, we are also going to discuss about the structure. Here it is translation. No, yeah. So this is what the structure of tRNA. Yeah. Oh, they have even given structure of tRNA. Okay, so this is what the structure of tRNA. So, here totally there are 5 different loops. What are those 5 different loops? Here I can name this as the first loop, this as the second loop, this as the third loop, this as the fourth loop, very important loop, and this as the fifth loop. Okay, so what is this first loop? First loop is what the amino acid acceptor loop. Okay, it is what the amino acid attachment loop. Acceptor end it is said as, or also it can be said as amino acid attachment loop. Attachment loop. So this is the place where amino acid will be attached. See, in this total entire structure, I'm saying, okay, it will be concluded as a box. This total structure will be concluded as a box, and only this will be written. Okay, this will be written on the top, like this only it will be. See, the total complex structure is what this box, this is what this tRNA, and then the attached amino acid is what this valine. Okay, and then this attached amino acid is what this valine. Like that only it will be concluded. So don't get confused. Okay, so here it is TYL. What is TYL? We will check out its name. What is over there? So it is THR. It is 309. Okay, so over there I think it is written as 309 only, or it is some other thing. Okay, 309 only. I think, I don't know whether it is 309 or something, but something over here. It is very messy image. Okay, so then the second is what? Ribosome attachment loop. Here is the place where ribosomes will be attached. Okay, and this is what the third loop is, which is anticodon loop, or also known as no-dot loop. Okay, also known as no-dot loop, and then this is what the fifth is, which is enzyme binding loop. Okay, enzyme binding loop, which can be, which is actually okay. So now we are going to see what are the chemicals that they have. Okay, so this one is not having any chemical. Okay, this one is not having any chemical, but this is what the first is, and this is the place where amino acid will be attached. Okay, now you might say, okay, so this tRNA is not the thing that is going to create amino acid. Yes, it is not the thing that is going to create amino acid. Amino acid will be already somewhere. Okay, that is just going to be energized. For example, if this is where the cell is, okay, and if this is where from the nucleus only tRNA will be released. Okay, so it will be released, and as it releases, there will be this particular amino acid. Okay, every tRNA based on this composition, based on this anticodon, based on this no-dot. Okay, based on this itself, here this amino acid will be very ready. Okay, this amino acid will be very ready. That is methionine. Okay, so here it is AUG. Yeah. So you have to convert it into there. It is anticodon. So for this, okay, I'll say very clear. Okay, so that tRNA is coming out, right? So the opt amino acid will be in the cell itself. That will be energized by adding of ATP. Then after that, this will be going and this will be combining with this tRNA, and that will be involving in the process of translation. Okay, isn't it very interesting? Okay, so what does this loop have? This loop has nothing. Okay, so this is okay. So what is this third loop known as? This third loop is known as external loop or external axon. Okay, external axon. Okay, so now coming to their enzymes. So this is having thiamine, pseudo-uridine, cytidine. Okay, what is this? This is having. See, you have to represent it as T. Okay, I will write in a different color. We will take dark green. Okay, so T like this. You have to represent. Okay, that means thiamine, uridine, cytidine. Okay, the cytidine, thiamine, pseudo-uridine. Okay, this represents pseudo. Okay, that small slash is there, right? Like this una, like this one is there, right? So that represents pseudo. So this is known as thiamidine, pseudo uridine cytidine, CYTIDIN. Okay, cytidine. Then after this external loop is there. Then yeah, here this is there, right? This is having DHU as its enzyme. So which is dihydroxyuridine. Okay, so dihydroxyuridine, and also this is the place where aminoacetyl tRNA synthetase is also there. Okay, this is the place where aminoacetyl tRNA synthesis is also there. Aminoacetyl tRNA synthetase is also over here. Okay, so here this is what UAG. Okay, this is what the codon is. So when you are taking the name for this you can see the name. Okay, see for UAG, yeah. So for UAG, where is UAG? UAG. So here this is what. Okay, this is what the stop codon is. That means this total process. Yeah. So UAG I should have got into my brain. Okay, so UAC, for UAC only we have to check. For UAG you should not check. For UAC only you have to check. So for UAC, it is actually known as trypsin. Not trypsin only, right? Or where? Yeah, it is known as, it is known as triptopane. There is no thing like trypsin. Where am I? In which world? Okay, so in triptopane. Okay, so triptopane. Triptopane. Then what about this anticodon? For anticodon you cannot check. Okay, for anticodon you cannot check in the table column. You have to convert this anticodon into codon. So for A what is complementary? U is complementary. For U what is complementary? A is complementary. For G, C is complementary. Don't put in place of U, don't put T, because there won't be any T at all. We are totally discussing only in mRNA. So for this there won't be any name, for this only there will be name. For anticodon or for no doc there won't be any name at all. That you have to be very very clear. Okay. So this is what the process is. Initiated. Yeah, so this is what the 2D diagram is. Okay, 2D diagram is. Okay, then what about 3D? Just imagine if 2D even is this much complex now, then 3D will be how much complex? I was just kidding. 3D is just L. Okay, that's it. L is its 3. Just this is what its diagram itself. Like this completed. L is what its 3 diagram is. It might be confusing, or you all might think that okay, I have said it wrongly. This is what 3D, that is what 2D. No, 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 no. This is what 3D diagram is. 2D diagram. Sorry, this is what the This is what the 2D diagram is. I'm very clear. Okay, this is what the 2D diagram is. 3D diagram is there, right? 3D is just L shaped. Okay, and this is only the 2D diagram. Okay. So after this, what we are having? We are having translation. Okay, sorry. Before translation, we have to see about mutation and genetic code. Okay, so mutation and then the genetic code. What is this mutation and this genetic code? Very simple process. Mutation and genetic code. The best example for this is sickle cell anemia. Where GUG has got converted into, okay, where sorry, GAG has got converted into GUG. Okay, so this is glutamic acid, this is valine. Okay, this is valine. It has got converted. Okay, so GAG codes for glutamic acid, GUG codes for valine. Okay, wow. Mutation, sickle cell anemia, GAG, GUG is there. Okay, now what we are going to see, this happens in chromosome 11. This is all not this portion. The previous lesson, chromosome. Chromosome 11. This is all previous lesson portions. Okay, in that one topic is there, sickle cell anemia. That portion. This is portion in sense that topic. This is okay. So here we are going to see about not that much, just frame shift mutation. Okay, what we are going to see is frame shift mutation. Which has insertion, that means you have inserted, okay, when you are actually inserting. Okay, so insertion, there are two types, deletion and then insertion. Okay, see, they have given us one amazing statement, that is great. What is that statement? Ram has red cat. Here has red cat. Oh, sorry. Okay, this is what the statement that they have given. What is this? Okay, so Ram has red cap. Okay, so something it is. But it is chromosome 11 that is it is hiding. Okay, so Ram has red cap. What we are going to do, we are first going to check about insertion. Okay, we are going to first check about insertion. First, I am going to insert a few letters. What am I going to insert? Let us first insert R. Okay, R itself. Any letter can be. For an example, consider this as a genetic code, this as a genetic code, this as a genetic code, and this as a genetic code. Okay, when you are considering all these as genetic codes. But the thing is, even though we don't have anything like R, we don't have anything like M, we don't have anything like H, we don't have anything like S, we don't have anything like E also, we don't have anything like D, we don't have anything like P as our nitrogen is bases, but let us consider that these four are codes. Okay, so when I insert R, okay, so when I, oh god, suddenly my marks came. How can I take it out? Okay, so neglect that part. Okay, so insert R. Now what am I going to do? I am going to do R. Okay, so let me insert it over here. That means Ram. Okay, before. Okay, wait. I'll insert over here in place of this R even. Okay, nearby this R even I'll insert. That means Ram has RRE it will be. Okay, then DCA it will be. Then it will be P over here. Okay, one is left out. That means all the amino acids that is before, okay, before the insertion are okay, that is nothing has happened. But after this insertion, see, everything has got changed. You cannot even understand. You cannot even notice that this came from here only. Yes, right. Now I'm going to insert two different letters. That means I'm going to insert R and E. R and E, which is Ram, same place, has RER, then RE, wait, RERE, okay, EDCAP is over here. So here also see how tremendous change it has got. Now what I'm going to see is, now I'm going to insert bigger. That means how it will be, Ram has big red cap. No change. Only this, the thing that has got inserted is as such. Okay, that is what is as such. Other than that, there is no other change. So that means these two, okay, these two that has got inserted, right? Those two are what causing the change. Causing the change. This BIG is not at all causing anything. Okay, not at all causing anything in sense it is causing something, but it is just a mere insertion. That's it. Okay, so this is what frame shift mutation. For that, this is what the insertion is. So my Ram, not my Ram. Ram has red cap. Okay, so yeah, when you do this, this is what will be happening in insertion. Deletion is also same thing, only kind of same. What is this deletion? Deletion is, okay, don't consider the color change between this R and this. Okay, so deletion, what am I going to, from this I'm going to delete one thing. So I'll delete R itself. I don't know what is my vengeance with R. So if I delete this R, what will be happening? We can say that as Ram has EDCAP. Okay, EDCAP, that 0, it's my marker, so don't consider that one. Okay, so then after that, I'm going to delete RE, so which can be said as Ram has DCAP. Okay, now I'm going to delete this totally, I read itself. That means Ram has cap. Now here it is totally unspecified. What color is it? Okay, we don't know the color of the cap. Okay, anyways, we won't do anything by knowing the color of the cap. Now we are going to jump into the concept of translation. With that, we'll be ending this total video. Okay, I was also thinking to say about lack of prawn, but that we'll be seeing in another video. That is translation. Okay, so this is what translation. Okay, so translation. So what is translation? Translation is the process by which mRNA or RNA converts, or RNA gives us, RNA gives us proteins. Okay, so RNA gives us proteins. Now we will check about, there are a lot fond of mosquitoes. My friends. Okay, my friends. First, why is this translation very, very important? First, what is this translation? See, proteins from RNA. Okay, so proteins are there now. From RNA, proteins will be created. This is what the phenomenon is. Actually, by using this process only, the expression happens. The total expression itself happens by just by this translation only. For an example, if I am having this particular color now, that is because the melatonin in my body are secreted in this particular range. That is why I am having this shade of color. If in case maybe if it were secreted in a large amount, then it will be blackish in color. Or if it were secreted in a little bit still more lesser amount, then it will be more whiter, fairer and fairer and fairer. Okay, that is what it is. Okay, so first we will be checking the phenomenons that will be happening over here. So first there is this cell. Okay, and then this is what the nucleus is. Okay, then the genetic material, the DNA is over here. I know, very, very big diagram, right? Thank you. Okay, so DNA is there. Okay, the genetic material is over here. This will be creating as RNA, and then this will be coming out. Okay, so as it comes out, there will be this particular, the required or the needed, not required, the opt. Okay, the opt amino acid will be over here. What gets is it gets ATP. It gets ATP, so it gets energized, and then it comes to here, and then it attaches. Okay, so this is what the first phenomenon that's happening. Okay, so it is being energized. All it gets is energized. This is what will be happening in a UK in sense, not in the United Kingdom. This is what will be happening in eukaryotes. Okay, then what about prokaryotes? Oh, shit. Thank God. Okay, in prokaryotes, this will be like this. Okay, we will conclude like this. Okay, there are no any nucleus, right? Like this, the genetic material will be. We have studied, right, in nucleosomes, nucleotids. It will be. Actually, they all will be positive proteins will be there and negative proteins will be surrounded. That is happening over here. Like that, only the DNA also in prokaryotes will be bundled. Okay. So coming to here, in large loops they will be in prokaryotes. Okay. So here what will be happening is translation and transcription. Okay. Transcription is happening. Then within fraction of a second, translation would have also started. Literally, they both will be parallel. That is what will be happening in prokaryotes. Okay, so by what? See, by enzyme peptide transformase. By enzyme peptide transformase. Okay, so first is charging of RNA. Okay, first this is what the phenomenon that's happening. Then after this, what's happening? What you have to do is you have to consider. So this is going to reach to this RNA. Okay, this is what going to reach this RNA. I will try to write as much as possible over here even. See, okay, that's not possible. This is not possible. Imagine this as the RNA. Okay, so this is the RNA. So we all know that there will be amino acids. And then there will be one big large head. Okay, one large big head, that is methionine. What is its name? I was the one who said it. Triphosphate. Yeah, it is triphosphate. It has triphosphate with it. So there is this head, there is this amino acid, and also there will be first UTR. First, in the very beginning of any RNA, okay, in any RNA there will be this UTR. So after some time, sorry, not after some time, after some space, okay, after this UTR region, there will be this starting region where the translation starts to occur. Okay, so this is the region where this, okay, where RNA will be there. RNA will be here. Like this will be. This RNA even is having 3 different compartments. First is entry point, okay, then next one point, then at last another one point. Okay, so these are those 3 different points. So we will be saying this as point A, P, and then E. Okay, so in this itself, this is the large subunit, this is the small subunit. Small subunit. Okay. One thing that I have to say is, in prokaryotes it will be different number, in eukaryotes it will be different number. We have seen that 30s, 40s, 50s, 60s, all these. That is what we are going to see. 30, 40, 50, 60, 70 and 80. Okay, so in this is there now. This is in prokaryotes. Prokaryotes, the small, this is what so small subunit is. For that it will be 30s. For prokaryotes it will be 30s. Okay, whereas for eukaryotes it will be 40s. So also same for here also. See, not same. It will be sorry. Yeah, correct only. So for this large subunit for prokaryotes it will be 50s, and then for eukaryotes it will be 60s. Okay, so they are total. When you are doing total, you cannot add them. You cannot add these also. What is this S? Actually, S is something that you can say as sedimentation. Okay, there's sedimentation gradient, I can say, or also I can say that as sedimentation coefficient. It is actually Swedberg unit. Swedberg unit or sedimentation gradient. Where, for an example, I can take this and this. I've dropped this in the water. This will be slow. This will be kind of fast. This will be sedimenting. Okay, but whereas this one cannot sediment that fast. It will be coming slowly. But when I combine these two, you cannot just ask me to add this, and they won't come in that their addition of their velocities, right? So they will be combining in a very different speed, because I think these two are vector. Both are velocity only, na? Okay, so. So yeah, this is for different, this is also for different. What are they? So here, this is for PK, and this is also for PK. This is for eukaryotes. UK, this is also for eukaryotes. Okay, UK. In the very beginning, the very beginning, the first amino acid comes. That will be like this, L shape, 3D. L shape, and that will be UAC. Okay, that will be UAC. This one will be having UAC inventory. See, I said here now UAG is here, okay, but UAC, that is what I'm going to talk about. Sorry, cut 20. Here in this place, AUG. See, AUG is here now. So like that, for each and every tRNA it will be differing. In one place it can be UAG, in one place it can be AUG, in one place it will be differing for every tRNA. Like that, the first tRNA that comes in the translation process, okay, the first tRNA that comes to the translation process will be UAC. Okay, why so? Because I have already said that the starting codon is going to be AUG. So here in the first in mRNA, what is going to be there? It is UAG will be going to be. Sorry, AUG is going to be there. AUG. For AUG, the opt complementary is what going to be? It is going to be UAC. That is what going to be complementary. So that is only will be there in the tRNA, and also that tRNA will be having its own amino acid. That is going to be methionine. Yeah, it's going to be methionine. You all know, right? That's going to be methionine. Don't think that. Wait, I said AUG. That means AUG will be over here. That means AUG will be over here, and methionine will be over there. That does not mean like that. AUG will be over here. AUG will be over here, and methionine will be over here, and the anticodon is going to be UAC. Okay, is going to be UAC. That is what it is. Okay, so first it will be entering into P only. Okay, so now it has got. Now after that, another tRNA will be entering. It can be any tRNA. You can take it as UUU. You can take it as CCC. As per your wish, that will be entering over here. By this one, it will be entering. Okay, over here it will be entering based on the genetic code that is over here. Okay, so after that, what happens is this will be this tRNA. This tRNA will be going here, and this tRNA will be exiting. Okay, this tRNA will be exiting. That is how the translation happens. See over here, even this is one column, this is one column, and this is also one column. What are 3 different columns I said? Nah, wait. This is what the first column is, where the entry column. Okay, this just comes. Then this is what the second attachment column is. Then this is what the leaving column is. This is what E, this is what P, and this is what A column is. So this tRNA comes over here, and then it just comes. Okay, this is where the first it comes. Okay, then after that it comes here. So this valine will be going and it will be going here. Okay, then they leave at E point. So tRNA come over. tRNAs come here. Okay, so after they have come here, then they will be giving their respective amino acid to this group. Okay, see this long chain is here, right? So to this protein, polymers of amino acid is what protein. So that is why we are saying that. Even I have not said in the very beginning. I'm very sorry. Polymers of amino acids. At least minimum 50 amino acids should be there. Then only it will be considered as a protein. Okay, so this comes over here, then this gives it, so it will be just elongating based on the principle or based on the function of that protein. This composition differs, and it will be exiting just like that. So it gives and it just goes. It gives and goes, and it will be preparing for the next translation process. Okay, so yeah. This is what we have to study. Then after this, there is only gene regulation. We will be seeing gene regulation afterwards. Okay, so that's it for today's video. We will be meeting in another amazing video. Bye bye.